I'm Chris Niogi. I'm a professor at UC Berkeley in the Plant and Microbial Biology Department, and today it's a great pleasure for me to interview my former postdoc mentor, Olle Björkman. Olle is a senior staff member, former senior scientist at the Carnegie Institution's Department of Plant Biology in Stanford, California, and we're here today to talk about his career in science. So, Olle, maybe we could start off by talking about the field that you helped develop, the field of plant physiological ecology, and maybe you could tell us what exactly that is.
Okay, I will try to do so, but I think I will go back a little bit further in time because the term physiological ecology had not been coined at that time. So, so in about 1956, I had taken a number of classes and degrees, all involving chemistry. And I wondered what to do next, what was a developing field that I might go into. And these were the days of Watson and Crick and just DNA becoming discovered, and I thought genetics would be a fine thing to continue with. So I moved from the University of Stockholm to the University of Uppsala, which then had a new genetics department that was housed in part in the Swedish Agricultural University plant breeding department. And the former professor there, his name was Göte Turesson, and he was a man who had coined the term ecotype or ecotypic differentiation. That means the microevolution, differentiation within species in response to the climate. In other words, plant adaptation that was genetically based. Now, I have a colleague there, Paul Holmgren, whose background was more in classical botany. And we were considering quite a bit how we were going to approach this problem with our various backgrounds and what processes and plant materials we were going to use for this. And we also needed the resources, that is equipment to grow plants under different environments, phytotron in other words, and we also needed other equipment to measure plant physiological processes, and photosynthesis obviously was the most that would come to your mind immediately if you're talking about plants. Since this process is a driving force, light is a driving force through this process, and everything else would depend upon it. So, with a lot of luck, there was a scout coming from the Rockefeller Foundation to Uppsala, and he came in when we were sitting at lunch one day, and he said, "What are you guys up to? And what would you like to do?" And we told him briefly. And he said, "That sounds good. Why don't you write me a proposal for tomorrow?" Well, we got it's okay, one page proposal. And wow. So we went ahead and wrote a one-page proposal. And some months later, it was approved. And that meant we had the resources to continue to do research for several years in this field. So in other words, I came into the field actually from the microevolution, although my background was very limited in that particular field. And to make a long story short, I ended up at the Carnegie Institution of Washington because it had a similar history when it came to studying microevolution, differentiation within species. The famous Timberline, Mather, and Stanford stations, among other things. So I came to join the group of experiment taxonomy. And after working there for a few years, and then 2 years, and having gotten a permanent position as a researcher there, and then I thought it was, and then I assumed the leadership of the group after Bill Heisey retired. And then I thought it was time to change the name of the group because it now was very much into physiology and ecology, of course. And my collaborator at that time was also Hal Mooney from Stanford across the street. So we decided we're going to call this physiological ecology. Not ecophysiology, but physiological ecology. And that became the name of the group. So going from there, so what is physiological ecology? Okay, it is essentially an all-inclusive bridging science that covers almost every aspect of plant physiology, genetics, chemistry, biochemistry, biophysics, whatever, bringing all these different fields together to solve the problem, which is quite simple but very hard to answer, and that is, how do plants adapt to extreme environments, or very contrasting environments at least. So I think that pretty much defines the term, and since that time the field has developed and probably means a lot more now than it did then.
So going back to your time in Sweden, were you trained as a botanist? Is that how you became interested in plants?
No, I never had a formal course in botany or floristics or anything. It was all chemistry and genetics.
Okay.
Of course, I had high school botany, but nothing at tertiary.
Right.
So that was self-taught. And in most cases, you can learn as you go, but if you have a reasonable background. Field.
So if you were interested in genetics and microevolution, what led you to plants then?
Well, what led me to plants was actually that I was in a department of genetics and plant breeding. So there were resources and history and a background there that I could benefit from. And also, the process of photosynthesis had fascinated me for quite some time, but taken from a biochemist level, not from a botany kind of approach.
And so those early experiments that you did in Sweden focused on a model system or organism that you helped to develop in those days, Solidago.
Yeah.
So how did you choose Solidago among the many plants that you could have investigated in terms of their responses to light and adaptation to light?
Well, my colleague Paul Holmgren and I made up a list of potential candidates for our research. It was a list of about 30 or so species. It included annuals, even Arabidopsis thaliana, which was very common and grew from almost all the way to the Arctic Ocean and Arctic tundra and down to the southernmost tip of Sweden. And of course, we also knew that it existed in more southern parts of Europe. We collected it in many places together with many other species. And the one that we settled for the main research was Solidago. It was not exclusively Solidago, but that was what came to be the main object of our investigations in Uppsala. Now, that had the following characteristics that were extremely important. First, it was a perennial. It was not an opportunist. It was a plant that grew in the same spot from year to year, and it had to endure the environment as it came.
Right.
And secondly, it had an enormous distribution all the way up again from the Arctic, but now up the mountains down to maritime environments, and even as far down as southern Spain and the island outside China that the communists were bombarding at the time, Quemoy. We didn't collect from there, but we did collect from many, many places. And at first, we concentrated on climate in terms of temperature and to some extent water relations. But we soon targeted in on light. Again, that was a primary component. And here we found a species that would grow in the open, in the Arctic tundra, where you had 24-hour light and intense light during most of the period when the plant was active, and also the floor of dark beech and oak forest, and where you might get about less than 1% of the light coming in on the canopy penetrating down to the floor. And also it had this very interesting or important rather characteristic, and that it would not become active until the canopy above it had leafed out. Mm-hmm. So we had a very nicely defined environment. Secondly, they were easy to hybridize with one another and extremely easy to clone. So they could vegetative cloning and get many individuals of exactly the same genotype. So that would enable us to do reciprocal and put them in different environments and comparing and so forth. So those were the basic reasons why we chose Solidago.
And from those early studies then of sun and shade ecotypes of Solidago, what were the conclusions? What were the findings from these experiments?
The conclusions were that there were obvious differences. If the plants from the exposed environments were grown in weak light, they would grow very slowly, if at all. And of course, we upped the light until we got the threshold. And but they, on the other hand, if they were grown under the highest light that we could achieve, they would grow very rapidly, and they would have high light-saturated rates of photosynthesis. The ones from native to the shaded habitats would grow very well under the low light, but obviously stunted growth when they were high light doses. And we discovered that the apparent quantum yield for photosynthesis, that is the amount of CO2 fixed or oxygen evolved per quantum absorbed, was quite a bit higher when they were grown in low light than they were grown in weak light, indicating that there was definitely some non-desirable effect on the system when they were grown in higher light. And conversely, we just just got the opposite from the plants, from the exposed environments. We proceeded to do hybridization and got F1s and F2s and F3s, and that turned out to be very highly variable. So we couldn't really pinpoint things from those hybrids. But we also found that was quite interesting that not very far from the deep shade in the forest, going out in a meadow, and we collected there as well, and there we got a variable response. So most likely it had some, there were more than one gene involved.
There was some hybridization occurring between nearby populations.
Well, later on, I followed up this work both in Sweden and in Palo Alto, or at Stanford. And then we went into doing things like measuring what was called carboxydismutase in those days, and now it's a Rubisco. And we found there that the shade environment clones, when grown in high light, would increase the amount of Rubisco a lot, several times. And whereas the I'm sorry, the ones from the exposed habitats, would increase the amount of Rubisco when grown at high light, whereas the ones from the shaded habitats had very limited ability to do so. And that was followed up by other things, so we now could balance and see what changes in the balance, what was then called the light reactions of photosynthesis, how they were balanced with the so-called dark reactions, which involved the Rubisco and those things. And we found that there were large difference in that respect. So, and of course we compared with species that would exclusively grow in the shade or in the sun, but did not have ecotypes that would do both. And there we found similar results.
So you didn't just stick with the one model organism, but looked at a variety of plants from different environments?
We did, depending on the question. Then, of course, we started to choose the materials that would be best suited for the particular questions that we were asking. And a real big advance was made when I went on a sabbatical leave to the Australian National University in Canberra, and I really didn't come there to do this kind of work. Most of the work that I was doing there concerned photorespiration, C4 photosynthesis and things like that. But I had the opportunity together with Keith Boardman's group, which included Jan Anderson, who became a close collaborator for quite some time. And now we had the possibility to start to look into the same question, but looking at very basic component processes and components of the photosynthetic apparatus. And it also gave me and there we used other species, and so Atroplex was one of the Atroplex patula was one of the major ones that was a sun plant. And another that we used collected from the Australian rainforest, the flora of Australian rainforest in Queensland. And one of those was Alocasia mycorrhiza, and which had enormous grana stacks, and we could do a lot of interesting work later on in the lab. So we could go beyond the differences that you would see within the ecotypes of Solidago. Here you could, go to the very extreme of the situation. So as far as our efforts to understand how this adaptation worked, this was probably the most important period, apart from the initial work with the Solidago. And then from this arose a new question, not only how could we explain that these shade plants had a better use of light than the sun plants, and why the sun plants could have a much higher light saturated rate of photosynthesis. But then came the question, why became these shade plants and shade ecotypes inferior when they were grown at higher light? There was some damage to the system, or some detrimental effect at least, which we would then use the term photoinhibition. And that photoinhibition actually got that term from Bessel Cook, who had been working on unicellular green algae a few years before. This was totally an in vitro system, not plants growing in the field or anything, but actually chloroplast fragments and things like this. But he found that excessive light would have a bad effect on photosystem too. And so I took that term from him and applied it to our effects. So this photoinhibition then became another field of study that we continued with for many years, and well, maybe proceeded with that a little bit later, your choice.
Sure. So I'm curious about the process of going from one area of inquiry to another. So you were led from studies of sunshade adaptation to photoinhibition. Was that just a natural progression as you described, or there were other things that you investigated along the way as well? And I'm just wondering how you picked the research problems that you investigated.
Yeah, it's a little bit hard to answer that question, but I'll try. Some of it is serendipity. So the plant part, of course, was observe the plants in nature, if possible. Getting some ideas of what responses might be or ought to be. Go to the lab, bring the plant material there, grow it under controlled conditions, see if you got anywhere close to what you were asking. Then, if you achieved results that seem to make sense and pertaining directly to the problem, you would return to the field to see if it really worked like that in nature. So when we did this, for example, we would find, apart from light, temperature is a very big factor, of course. And so we decided to do research in the floor of Death Valley in the summer and see what plants were growing there, if anything was really growing at that time. They obviously were there, but were they really active? Did they do photosynthesis and things like that? And in order to find out, you have to go there, and you have to share the misery with the plants. Okay, so we had to construct a mobile laboratory and bring it to Death Valley, among other places, to do this kind of research. And then while you were there, you discover new things. When there were new questions that came up, and you got a different perspective in terms of the interaction between high temperature, high light, and water relations, and so forth. And you then have to approach this problem trying to make an analysis possible. You have to do it factorially, I believe, okay? But you also have to see when you're putting the different factors together that it will work the way that you thought. And so that brought us into, other than light, of course, C4 photosynthesis because of its importance in, I'm not talking about the ecological aspects of C4 photosynthesis. We had much earlier worked on the more physiological aspects of the process. So in that valley then, we got new ideas and we started new programs with transplant that had to do with light and light interactions with temperature. And then we had to find out a rather considerable program for 4 or 5 years involved adaptation to temperature. So that, but never in a vacuum. So we never studied photosynthesis in a vacuum. It was always together with a plant and its environment. Okay, and so what happens then when you start to choose different species that will suit the immediate problem that you're working on? You study this plant in the field and you become fascinated with some other aspects of it. So, for example, when we come to the interaction between food inhibition or excessive light and drought, the rationale here is that when you have drought, the plants will start to close their stomates in order to avoid excessive water loss, passive water loss, as water vapor through the leaves. And that would in turn lower the rate of photosynthesis and therefore increase the excessive light. So even though the light wasn't excessive before, it now becomes excessive. So you have to look at it that way. Now, drought is a very difficult factor to control because it very easily goes from nothing to a dead plant. Okay, yeah. So it's very hard to keep it constant. And it occurred to me at the time that perhaps if we're talking about physiological drought, we could find a more constant one by studying mangroves, because mangroves are essentially trees that grow in the ocean and has a very high salt concentration. Therefore, there's a high osmotic stress in there. So that gave me a chance to have a more constant stress factor. So I went to, in 1983, I went to North Queensland, just close to the Great Barrier Reef, to study the mangroves with a mobile operation there. And from that emerged that these plants were under incredible stress with a highlight tropical sun on them at all times. And I got a severe food inhibition effect on mangroves when their leaves were in highlight, but the drought or the salinity stress had no effect at all on them when they were in shaded situations. So here you had obviously no bad effect of the high salinity, other than when the light was high. And then, but then came a new thought from this. Maybe it wasn't all damage. Maybe actually in these mangroves, I start to suspect very strongly that something I had observed a bit early under circumstances, that there may be a way that these plants actually shunted off some of the light that became temporarily ineffective in using weak light. But over time they would recover without, and actually there may not have been any direct damage to the system. Okay, so coming back then to California, to Carnegie, bringing back with me a number of mangroves that we then grew under controlled conditions, and that's from there on. These were used in some subsequent work that also came to involve photochemical quenching and the carotenoids and the zeaxanthin, vioxanthin cycles. So, but I have to go back a bit in time now, and this was just how the interaction between different factors and how that spurred new ideas and questions that we needed to solve with it.
Right. So the work in the field really was a major driver for you then, in terms of noticing things that were occurring in nature and giving you new ideas for experimental directions.
It was on the whole. I probably didn't spend more than 10% of my time in the field, but that time was crucial to the how we would approach and design the experiments that we'd be doing in the lab.
But there's probably a good lesson for people like me who probably spend too much time in the lab. A lot of good ideas can come from just observing what's happening in nature.
Yeah, it's a main investment, and it was because you need special equipment and you need very hardy collaborators. And so the planning of the field research takes much longer than the actual stay in the field, of course. And we also built or established transplant stations in different places, including the floor of Death Valley and a cool, miserable place up at Bodega Heads, north of the Tomales Bay there. Yeah, I think it was important. I think it was quite important.
So in terms of being able to do experiments in the field, it seems, well, in the lab you have controlled conditions, and it's relatively easy to have the equipment at hand there to do very precise measurements of photosynthesis and such things. But maybe you can talk a little bit about the logistics or the difficulties of trying to do such measurements in the field in places like Death Valley.
Yeah, the logistics then would be a lot harder than it is today, because you had to construct the equipment that you were to use. You had to have a data acquisition system of some sort. And initially, of course, in the late 60s, they were not computer-based. You had to get a rather clunky kind of equipment and recording systems with lots of paper and points. And it would also, the equipment was also much more heat dissipating. A lot more heat would be dissipated from the system used for measurement, which is hard when it's already extremely hot outside. So that means the generators have to be larger and suffer more. So it was a big deal. You had to construct a mobile laboratory that could take these conditions and still be not too large, so you can get into the sites where the plants were, and without causing a lot of destruction of the habitat. So we actually had the same mobile laboratory together with Hal Mooney in Death Valley I used the same one in the redwood forest to study Oxalis oregana in about 1980 with Steve Powell's. They very different situations, but by that time, 10 years later, that is, it now did have a computer the size of a washing machine and things like that. But and we could do a lot more. But technology goes so much for mobile measurements or photosynthesis and lots of other things. You know, all the fluorescence measurements they can do now, and all of them in combination, light-induced absorbance changes and things that we could do in the lab quite successfully. But it was a fantastic challenge to try to do anything like that in the field.
So I think one thing you're known for is the creativity in your experiments, and I think the experiments with oxalis that you mentioned in the redwood forest is a great example of that. Maybe you can just describe that experiment that you did with Steve Powell's.
Well, Steve Powell's came out as a postdoc, and he had been working with Bayer Osman in Australia and had got into this thing with photoinhibition, and so he wanted to come to work with me, and we did. And for the most part, we did not work with oxalis out in the field, but rather other important developing fluorescent measurement systems and things like this, and working on other species. But a couple of years earlier, I got intrigued by this Oxalis oregana that grows in the redwood forest mostly, and is native to the coast here, related to the shamrock of Ireland and so forth. And they all share a very interesting response, namely that they can adjust their leaf angle to maximize light interception when the light is low, and to completely avoid it when the light is high. So when they can move their leaflets, so like this, any angle, and they can follow it as the weak light comes in, maximize interception at all times. Then when you suddenly get a sun fleck, within seconds, they fold down like an umbrella, like this, and stay parallel to the sun's rays. And of course, this is a very interesting mechanism on a different organizational level than the organizations of the photosystem 2 units or the Rubisco or whatever, but it was still an extremely effective system. And here we also had the opportunity to compare these plants with shade plants from a similar situation, but were unable to do these movements. And so we decided we're going to go out with a mobile laboratory and do measurements in the field and see how much photosynthesis would these do under the various conditions, how fast were the responses in the field with real sun flecks, and we had an enormous lot of fun doing these experiments. And then we could come back to the lab and reconstruct things, so we can now build so we could have real sun, but we could now filter out different wavelengths and do a lot of experiments that were very hard to do in the field. But that would then tell us a lot more about the mechanistic aspect, what were the sensors involved, and what was the ion fluxes and things like this that caused the leaf movements. We worked on other forms of leaf movements, but yeah, it was something that was fun and added to the the whole bag of tricks that the plants can do to to either maximize or avoid light interception.
So you mentioned this way that plants have of shunting some of the excess light that they absorb in a way that is protective rather than damaging to the plant. Maybe we can talk about how you got into research on that.
Yeah. Well, of course, I have that longer background about the photoinhibition and actual damage that obviously did occur. But to what extent was this a reduction in the quantum yield of photosynthesis caused by a real damage to the system, and to what extent could there be involved a mechanism that would render the excessive light, or that would shunt the excessive excitation energy away from the photosystem to reaction centers. And this is going back to when Steve House came to the lab in 1979. And we constructed these fluorescence measurements, extreme, mostly for liquid nitrogen temperature measurements. And we got, or I did, involved a little bit behind the theory of fluorescence quenching, which had been studied in the lab, mostly in particles, in vitro preparations by Warren Butler in Southern California, and he had come up with beautiful equations where you could distinguish between fluorescence quenching, that is a massive reduction in fluorescence emission from the chloroplast, whether that would occur in the antenna, that is in the light collecting system, or at the reaction center. And using his equations, we could then develop a hypothesis how this may turn out. So what we found then was that there was a component that would not relax over minutes. It would take more like hours or days, or even longer, for a component to relax. And could that be a photoprotective thing, or was that just a damage to what would probably be what was then thought to be the D1 protein? And so we tried to design a number of experiments to resolve this, and I think we were partially successful. But then we let that lie for a while, and then I went to Australia for this mangrove work and came back in '83. And then Barbara Demig from the Würzburg group in Germany had asked to join me. She was essentially ecologist, and she was interested in mangroves, and she wanted to do research on the mangroves, and that I was bringing back, so that would be a good opportunity. And of course she did. But during this work with the mangroves, she continued, or we closely worked together in the lab, but we tried to explain this fluorescence quenching that we observed. And then there was another form of fluorescence quenching that had been observed, I think, first by Krause in Germany, which was a fast relaxing component, and what's called energy quenching, which was presumably caused by the development of a protein gradient across the membrane. And it took quite a while to separate these things out because it would be very hard to explain how such a proton gradient could survive for even minutes, or not to speak about hours or days. Okay, and there obviously must be two different forms of quenching. Well, Barbara went back to Germany after a few years, and then she sorry. Before that, before she went back to Germany, she came up with numerous ideas each day. What the next experiment might be, and how to go about it, and it was my job to sort out these things, and both for practicality and whether it would be realistic to do it within the time frame that we had. And one of them, and I don't really know the whole background for this, but she said, well, maybe this conversion of vioxanthin to zeaxanthin is involved in this somehow, because they have similar kind of responses over the short term at least, and it made me continue to do some work on that, and digging up the literature on carotenoids instead what we had been doing. She went back to Germany and then proceeded to do some experiments with the Würzburg group that had been very successful in developing an equipment measurement system for so-called room temperature fluorescence quenching and so forth. And then pretty soon, on both sides, both in Germany and in my lab, this relationship started to become more and more obvious, all right, and that there must be something that was a unifying factor. So that's, I think, how we got now into the more mechanistic aspects of photoprotection, how it would work. Now, from there on, you know a lot about the history.
Maybe we could talk a little bit about your style as a scientist. So you've mentioned some individuals that you worked with over the years, and it seems to me that you have always worked closely with one or a few colleagues, rather than having a very large group or a very large lab of people focused on the questions you were interested in. So it's quite a different style from what many people nowadays do as scientists, and maybe you can comment on that.
I think it mainly because I'm a bench, hands-on scientist. I like to do experiments, and so maybe the most important factor is not the whole thing, but the most important factor is that I want to be involved in carrying out the experiments, because that gives you the joy of discovery. Whether it's new or not, you find out later, but it's the joy of discovery. You see it right there and then. And if you have a small group and you're working closely in the lab with those people, you're on top of it immediately and share the joy. Okay. If the group becomes too large, you become an administrator of the research, and also it becomes more costly, and you have to rely more and more on outside grants and spend more of your time writing grant proposals rather than conducting the research. Right. So But I think I have worked with larger groups, and successfully so, temporarily, you know, for up to a year or so, with one group and then different group at another time, and I enjoy that a lot too. But those were mostly, these were never top-down kind of things. Those were association of like-minded people who like to get together to do things to solve a problem, okay, and with who also did have independent sources of funding, so that didn't become a problem. But on the whole, I just think that I just enjoyed a lot sitting down with my collaborators. If we were 2 or 3, even better, because the communication was direct. We could come up with wild ideas, and there was very little that you might get in a larger group when some of them might feel that they are subject to ridicule or something like that. But with a smaller group like this, you can really just throw out any idea you have, and I enjoyed that a lot. That give and take kind of thing, very enjoyable. And then I really like to design equipment that would be suitable for my experiments. So I never acquired a piece of equipment and find out what you can do with it. I always had a problem if the equipment to solve that problem was available and I had the money, I would buy it. But if not, I would construct it. Okay, so, and that construction part was also a lot of fun. The building of the equipment made you look a little bit closer to the problem also, and you became, I would say, more careful with the amount of, or the number of experiments you were doing. You design the experiments more carefully, so you didn't end up with just a massive amount of data that you must have spent months processing afterwards, right. And that may be all nonsense. Okay, so I think that was a very successful way of an approach at the time. I kind of doubt that it can be done today, but even if you really wanted to, it would be very, very difficult to do in most situations.
Yeah, I think nowadays there are a lot of distractions in a career as a scientist that pull you in different directions and away from the lab bench. So it's very hard to maintain the kind of focus that's necessary to keep doing experiments throughout your career.
Yeah.
So do you have any secrets for how to maintain that focus?
Just say no. Well, you have to do your share, but it's easy. If you gain some level of prominence in your career time, you get invited to many meetings, and then you will get invited to different panels, and then you will be invited because you have been invited to other panels and your name is on lists. And now you can probably spend more than half of your time just doing this, and it will just, you know, increase until it's no longer feasible. So I think you have to be jealous of your time here. How much time can you really spend on doing the research? And you have, of course, you have to go to some conferences and things and exchange ideas with others. There's no doubt about that. But mostly I prefer that, again, to do it in small groups. I tried to give priority to meetings that have fewer than 150 participants, rather than the ones that had several thousand, right, or more. And so that I don't think I have any secret formula that but just being able to say no to some flattering invitations, yes, is an important.
So maybe we can talk a little bit about the environment at the Carnegie Institution. When I was a postdoc there, I noticed sort of what a unique place it was, and I heard a lot of stories about how things worked there over the years, and maybe you can comment on that a bit, how that environment shaped your career or helped you do your science.
Well, so I came to Carnegie in January 1964, and I retired in 1999, so that's a lot of years in between. And that involves 4 presidents of the Carnegie Institution, and three directors at the Department of Plant Biology. And it involved a change in the funding structure from going to the style of 1964, when the National Science Foundation was fairly new, and when we at Carnegie were not supposed to accept federal grants. And there were 2 reasons, I think. The primary reason might have been that the former president, Vannevar Bush, was also the main person behind the formation, the establishment of the National Science Foundation. And the whole idea of federal funded research came from him. So it was considered unethical for the Carnegie people to go for that money. And also, I think the Carnegie in those days were guarding themselves against any external influence on the principles of Andrew Carnegie. That was that the institution, Carnegie Institution, should conduct basic research to the benefit of mankind. And there was an emphasis on basic, not applied. Okay. And I think what I thought is that they had external funding at the time, that it would be money driven, or that the kind of ideas, what fields of science ought to give priorities and so forth, would somehow diminish the freedom of the Carnegie scientists. Well, this was a bit of a problem, because post-Sputnik there was a lot of federal money going into research, so conducting research became more and more expensive because of equipment and other things that came. And so by about 1969 or 70, I found myself in a dilemma. We want to have a mobile laboratory, we wanted to build equipment, but eventually I got around that and got permission to get money for major equipment, but not for any running expenses or from any salaries of any sort. No one should be on soft money, was the idea at the time. Okay. So that was a happy compromise, because over the years this changed, that it would be impossible not to have federal grants to run any sizable scientific program. So So there were good points and bad points with this, and I think that also was reflected in the way things were carried out. The strength of the Carnegie Institution in 1964, the freedom, total freedom, very little duties outside of doing the research, committee work, all of that. And I would say an encouragement to go for blue sky research, high risk stuff, then, you know, that what you do. But as long as was new and innovative stuff, then go ahead and do it. And that was, you did not have to get your proposals reviewed by anyone. You could just go ahead and do it. Okay? So that was certainly a factor. Well, as we progressed through time, very, very important factor. Carnegie also had postdoctoral research fellowships that were quite generous, at least very liberal as comes to terms of using them. So you could have 1 or 2 postdocs at Carnegie fellowships, and they could request to come, or you could invite them, or there was no administrative things involved in this. Very easy to go about it. And if you were at a meeting or a conference or something like that, and you met someone who seemed very interesting to work with and so forth, you really could just go ahead and invite them. You know, this was quite something. And over the years, of course, the institution grew a lot, or at least the department grew a lot, and that the number of fellowships that the institution could provide, of course, would not be enough at all. So it was diluted out, let me say, mostly, and as the department grew. I think the very best years, as far as a combination of the funding situation and ideas and vitality, was from about 1969 through 72, 73, with big things coming together in a lot of areas of photosynthesis that we were doing, and ecology, and all the meshing with C4s and photorespiration and so forth. And where people would come in and maybe stay for a few months or for a year or 2, and then regrouping and having a lot of fun together, and then exchanging. So, for example, with a Canberra group, that was a give and take all the time, and also to some extent with the German groups and even the Swedish ones. So there was a more informal and immediate contact among the scientists than we could sustain in the long term. That's not to say that it wasn't really good all the time. Sure. You know, I'm just saying that this is, and it was still very good when you were there.
Just one other question related to coming to Carnegie. Did you ever imagine when you left Sweden that you would be coming to the U.S. Permanently?
This was a 1-year fellowship. And I was well prepared for it, though. But I really had no intention to stay more than 1 year for this. But it turned out, a stroke of luck, that the U.S. consulate or embassy called me when I had applied for an exchange visa and said, we think that you should apply for an immigration visa. And I said, well, I have no intention to emigrate anywhere. But we looked at your situation, and here you have a family, 2 kids and a wife, and if you can become a resident, your tax situation is much better, and so forth. And who knows, these were the Fulbright fellowship days, all mine wasn't, but the idea then was to prevent the so-called brain drain from Europe, so they had a 2-year limit. If you had an exchange visa, you could not stay for more than 2 years at all. So they said that it's better to keep it open. Okay, so it's kind of a strange thing to become solicited to do this, but I did, and so when then the opportunity came to stay, there were no hindrance from that side that people would encounter now all the time. Okay, and at that time there was also this idea that the ethnic composition of the country should remain reasonably stable. So a country that had a huge immigration, like Sweden, had a very high quota, and people like India had a very tiny one. Okay, so there were no hindrance there. Kind of funny. I'm bringing this up because I went to a meeting with the American Academy of Sciences at Stanford that John Hennessy, the president, was guiding, and he said that one of the real problems today is that you bring these guys over mostly from Asia, but also other countries, and they do their PhD here and they stay at the university, great cost to the country for 5 or 6 years, and then you force them to leave the country and they take all it with them, and here we get in a very bad situation. And in order to Congress could actually make a very, very important decision just by eliminating this rule. And so I'm putting this in a little bit in context here. I think it's important.
Right. Yeah. So over the years you published a lot of papers. Many of them you were the first author on because you did the experiments yourself. Is there one paper that really stands out from your career that was your favorite or a particularly memorable result that you published?
Well, of course, the 1963 paper in Solidago will stand out over time. And then I think the other paper that I would say number 2 in jumping a number of years later would be the papers for the Canberra conference on photorespiration and photosynthesis in 1970. And I think the last paper, probably the one with Lee and you and she and myself, Grossman, yeah, about the role of PSBS. I think that was, those are the 3 papers over a long time that I would immediately remember. Right?
Well, it's interesting that your career is kind of bookended by genetics experiments. You started off working on Solidago, wanting to use that as a genetic system to learn about sunshade acclimation, and then when I got to work with you at Carnegie, we did a lot of genetics together.
Right. No, you brought that aspect in, or at least techniques in, both with Chlamydomonas and Arabidopsis, that we were able to benefit from that. So that was a very good combination of the fluorescence measurements, imaging systems, and all of that together with your molecular biological techniques that in combination that would enable us to get the right mutants.
Right. That was another good example of where building the equipment was a key aspect of the project. The video imaging system that you built was really, without that, we would have been screening one by one or something.
Right, that would have been very hard.
We would have eventually found the mutants that we were looking for, but it would have been a lot harder, that's for sure.
Yeah. Well, that was a lot of fun that time too. I really enjoyed that a lot. So what do you see the field going from here then? Not physiological ecology, but the photo protection.
Well, I think things have progressed since I left Carnegie quite a bit, and we've learned a lot about how this shunt or safety valve for photosynthesis works in plants. And I think a new area for me, at least, is again kind of looking beyond the one model organism, Arabidopsis, and looking at other photosynthetic organisms out there in nature and trying to learn about what strategies they have for dealing with excess light and for protecting themselves. So we've recently started to work more with the algae and kind of returned to some of the Chlamydomonas mutants that I actually isolated when I was working with you. And we've learned that algae do things a bit differently from plants.
Right.
Same overall goal, but the machinery is a bit different. And I think that kind of comparative approach is really instructive. And that's what I would like to continue doing in the future.
So we're kind of closing the circle then, from about 1959 to
2011. So maybe that's a good place to end our conversation. It's been wonderful to talk with you about your career and to learn about how you did things along the way and some of the really cool results that you obtained during your research career.
Well, it was a privilege to be interviewed by you. Enjoyed it too.
Great. All right.